Rapid pesticide screening can be completed utilizing high-resolution mass spectrometry and the DART ionization source. Prior to this analysis, scientists at the FDA collected samples on small polyethylene sponges. These sponges are now positioned in the new flow-through screening carrier. Multiple sponges are applied with one space between each. With every other hole occupied, samples are effectively isolated from each other. A cover plate is attached to hold them in place, and the fixture is applied to the linear rail of the DART SVP. Each sample passes in front of the DART source, with ions being formed as the gas passes through the screen. The vapor interface collects the ions and carries them into the mass spectrometer for analysis. The time scale of this experiment is accelerated here. In reality, each exposure is about 2 minutes. After the desorption from the sponge has been completed, the module is advanced to the position where there is no sample in the way of the DART gas. The gas flows through without interacting any surfaces, and the DART gun is allowed to re-equilibrate. A partial mass spectrum is shown here to demonstrate the utility of the method. Here, 8 separate pesticides, which would appear in this small 3-mass unit window, are clearly evident. The entire mass range is not shown due to the fact that too many ions would be evident. Mass chromatography was used for each ion to determine the signal to noise for the peaks, and all of these ions showed good signal to noise. The utility of DART for screening literally hundreds of pesticides can be demonstrated using this method, which has only begun to be developed.